Hello everyone, and welcome to our online lecture series for Bio2. Today we take a look at viruses and their position on the tree of life. From here on out in the class, we will be discussing different lineages on the tree of life in hopes of better understanding and surveying the biodiversity on planet Earth. So let's go ahead and get started with the very root of our tree of life. From different lines of evidence, including experiments, geological studies, surveying other planets, we know that Earth, largely in its beginning, was a water-covered planet that did not have oxygen, or was anoxic. And it was under these, largely aquatic, oxygen-free environments that life evolved. Now, the evolution of life began with the most simple organisms. The prokaryotic bacteria are a very, very diverse lineage that we'll spend a bit of time on next time, looking over how diverse this group is, what are their different modes of existence, and some of their basic building blocks. These simple prokaryotic life forms, some of them stayed this way and have continued to evolve and live on planet Earth. Bacteria are a regular component of our lives, involved in everything from food making to diseases. But some of them evolved a more complex organization internally. Those groups are the archaebacteria and the eukaryotes. The eukaryotes being those organisms with membrane-bound organelles, like a nucleus. But wait, this is a lecture about viruses. So where on the tree of life are actual viruses? Well, they're not. They're not on this tree of life. We have hypotheses about what they might have evolved from or how they came to be, but fundamentally, viruses are not alive. They need to replicate in their host. Therefore, they're not technically classified on the tree of life. Whereas you may have thought of viruses as their own independent living thing with the goal of getting you sick and moving around and getting and involving and getting more people sick, I really want you to purge that idea from your head. Instead, start to think of viruses as these strings of computer code that copy themselves and fill up your hard drive. Or something like a catchy song that keeps playing in your head. They have no purpose. They themselves are not trying to gather more resources, but they just happen to be this combination of protein protection and genetic coding nucleic acids that go around from cell to cell, cells that they can get into, and just self-replicate. And in achieving their replication inside a host, they oftentimes lead to side effects. We as humans call those side effects getting sick. Here is an image of one of the most probably well-recognized viruses nowadays, that being the SARS-CoV-2 virus that leads to the side effects that we call COVID-19. So and the more I study biology, I will admit to this. The more I study biology, the more appreciation I have for the fact that viruses are not actually living things, but just pieces or fragments of macromolecules that can self-replicate. They don't know what side effects they're causing. They don't care what side effects they're causing. They're trying to find ways just to copy themselves. So don't think of them as evil. Don't think of them as obviously good. Just think of them as these catchy songs that go from here to there, and in doing so, maybe block up the system and actually cause failure of the system. Just like filling up your hard drive actually causes, your computer to perform slower and up until you delete and release some space. Just like I was finding out in compiling a lot of these lecture videos, once my computer hard drive got filled with lecture videos, I started compiling slower and slower until I realized that it was actually my space filling up. So I cleared some of them off onto an external, cleared off some of the space, and was able to get back to functioning normally. Side effects of self-replicating materials. So hopefully with that introduction to how to frame your thinking about viruses, we can get to talking about what they actually are and even looking at some either digital models or some real pictures of viruses. So fundamentally, what is a virus? It is something that can infect or enter literally anything. Meaning that there are viruses that can infect bacteria. There are viruses that can infect eukaryotes. And anything that they can get into, they will try to self-replicate. So viruses can attack or enter any branch of the tree of life. This means that viruses fundamentally are highly variable. Incredibly highly variable. We actually first discovered viruses when a scientist was putting pieces through a porcelain filter, trying to get rid of pretty much anything living, and for this tobacco plant mash. So he got rid of all of the bacteria in the porcelain filter, but then found that that mash could still pass on the disease. His conclusion, therefore, was that there was something smaller than bacteria present in that tobacco leaf that could pass on this infection. And the disease that he was working with was tobacco mosaic virus, or TMV. And we've done a lot of work on TMV. This is what it looks like under an electron microscope at 160,000 times magnification. That's incredibly small. So viruses are considered submacroscopic, meaning that not even a light microscope will allow you to see viruses. They're tiny. The singular viral particle is called a virion, and its size ranges from 20 to 1,000 nanometers, meaning that they are just incredibly small. Now, we have some ideas about their origin, of some hypotheses about their origin. These hypotheses are incredibly difficult to test. So fundamentally, their origin can be summed up as who really knows. But the three leading hypotheses are that viral origins are either regressive, meaning that they simplified from cells, meaning that they once were living things or living cells that then lost their ability to self-replicate and instead now replicate in other organisms. So you can imagine fragments of DNA and protein joining up together to accomplish that feat. That they are progressive in origin, that they are just escaped DNA from a cell or something of the sort. So they happen to be pieces of DNA that escaped from the cell that then got bound to proteins out in the world and then started replicating in other hosts. Or that they themselves are self-replicating DNA. Oops. Like the transposable elements, those highly repetitive components of our genomes and other genomes that really just go about copying themselves and have comprised, in some, for example, plant species, upwards of 80% of the genome. Not very difficult to imagine that something that just copies itself till it takes up 80% of all of the DNA in a 3 billion base pair genome to also snap off from the genome and continue to self-replicate and then replicate in other lineages as well. So, don't want y'all to focus too much on this because we have some very vague hypotheses. But what we do know about viruses is that they do keep evolving. So while we don't know their origin, we know that they have their own viral tree of life virus? Tree of virus. In terms of viral structure, this is incredibly important. Viruses are not cells. They are instead comprised of up to 3 components. The first component being some sort of nucleic acid core. That nucleic acid core can be made up of either RNA or DNA. More on that in a minute. The second, always there component, is a protein capsid. The protein capsid being a coat on the outside, the protective layer that keeps the DNA or RNA safe, but also actually oftentimes provides a scaffold for the DNA or RNA to hold on to. The DNA oftentimes weaves itself through the protein coat, so that way it is protected and not going nowhere. I find that fascinating. And then third, and this one's kind of a maybe sometimes, they have an envelope made of lipids on the outside. Oftentimes, that envelope is actually the host plasma membrane, a component of the host cell. Kind of wicked that it actually takes a piece of the thing that it's trying to infect in order to keep itself safe and then move along into the next quote-unquote victim. If that isn't a wolf in sheep's clothing, literally, it's taking a piece of the plasma membrane or skin of the cell in order to hide itself. I don't know what is. That's definitely a wolf in sheep's clothing. The reason for these different structures are quite simple. The nucleic acid core makes the protein coat, and the protein coat protects the nucleic acid, plus allows for entry into a new cell. Then the nucleic acid core copies itself, and then goes and makes more proteins to continue this process. See the whole self-replicating thing in the host, which makes it really weird, very just Selfish, greedy, not caring what's going on, and just copying itself. These viruses tend to come in 3 broad classes of shapes. Those that are helical, you'll notice yellow protein, red nucleic acid. It oftentimes in helical viruses winds along the side of the helical virus. The tobacco mosaic virus is a good example of one of these helical viruses. A lot of plant viruses, most plant viruses, are helical. Before this lecture, you may not have known that plants get viruses, but oh boy, they do. We even discovered viruses by working on plants. Gotta say that as my PhD is in plant biology. The second shape is icosahedral, some sort of, think of this as a multi-sided dice. For those of you who may have partaken in dice playing games as a child, like Dungeons and Dragons, think of this as a D20 or even more sided dice. The SARS-CoV-2 virus is an icosahedral virus, many different faces to this viral shape, with that very particular spike protein sticking out on the top of it. And then the last is we don't know how to describe it, so let's just call the third category complex. I'm sure as viral biology gets more defined and we study viruses more, there may be more additions to viral shape and fleshing out of complexity. But for now, we have those that are helical, those that are icosahedral, and those that are not those two. So those 3 shapes can also be broken down, or in addition to breaking down viruses based on shape, we can take a look at some of the fact of whether they are naked, meaning no envelope, or enveloped. And that one makes sense, right? So you have some naked icosahedral viruses like Hep A or polio. And then you have some enveloped icosahedral like herpes, yellow fever, or HIV-1. And then, of course, you have the same with the naked and enveloped helical. Enveloped helical being our influenza viruses, our mumps viruses and measles viruses, as well as rabies, quite spooky. Thankfully, we have addressed mumps and measles through vaccines, so hopefully you have not had to worry about these infections in your own life due to vaccination. And then we have those that are the complex with many different proteins, combinations of these different types, like the smallpox virus. All right, so now that we know what a virus is made of, the nucleic acid core and the protein capsid with sometimes a envelope on the outside, we need to talk about how a virus gets into different cells, because that entry is absolutely critical to its propagation, to it copying itself. Viral entry into a cell relies on one of 2 different methods. The first is receptor-mediated entry. And the second is membrane fusion. In receptor-mediated entry, you have some sort of plasma membrane receptor, so a protein on the plasma membrane of the host, that will bind to one of these viral proteins. These plasma membrane receptors are oftentimes glycoproteins, or sugar and protein combined together, some sort of signaling molecule. And that combination of the two begins the process of endocytosis. or the bringing in of material from the outside of the cell. So essentially, the virus has a key to get into the cell. It finds the particular lock, with your plasma membrane receptor being your lock. And then once it finds that lock, it convinces that lock to open the door and it gets in. In the case of the very popular, or well-recognized, SARS-CoV-2, the key, the viral protein that allows the SARS-CoV-2 virus to get into our cells, is this spike protein. And the spike protein will bind to a very particular receptor on our cells called ACE2, or angiotensinogen converting enzyme 2. Angiotensinogen is a hormone that regulates blood pressure. So there are a lot of different tissues in the body that have a role in regulating blood pressure, like all of the tissues in the lungs, like all of the blood vessels, liver, kidneys, all kinds of different things. Hence why we see so much diversity in where SARS-CoV-2 was infecting. Anything that has that receptor can act as the lock for the key for that spike protein. So that's the general idea behind that receptor-mediated entry. In terms of membrane fusion, well, that is a lot more simple. The virus has a membrane that oftentimes derives from the host, and because it is both a membrane and from the host, they can fuse together and the virus will just waltz in. Essentially, the membrane becomes permeable to the virus. Wolf in sheep's clothing. Always think of that. So what are some examples in addition to SARS-CoV-2 and its spike? We also have human immunodeficiency virus, or HIV. HIV has proteins on its side that will bind to a combination of our proteins. HIV needs two hands to get in, so it is looking for two receptors. The first is a protein called CD4, and the second hand or entry point is CCR5 or the human receptor CXCR4. Very memorable names, I know. We'll stick with just those names. But note that because HIV has these two hands, it needs two plasma membrane receptors. The crazy thing about human populations is that not every population of humans has that second hand. So there are some populations of humans that are naturally immune to HIV because they have one of those receptors that HIV will need to get in. So HIV will be floating in the bloodstream, bind to one of those receptors, but without ever binding to the second one, the lock will never be open. So there is no lock for the key of HIV. Granted, this is an incredibly small population of humans, focused around the Ural Mountains. Which is in like Western Russia. And we've seen some really, really bizarre rates of HIV disease there because it's so incredibly low, despite the fact that this area is generally fairly poverty-stricken and has extremely high use of intravenous drugs. So in areas where we see high use of intravenous drugs, HIV transmission is much higher. But despite the fact that these individuals may be intravenously injecting themselves with HIV by sharing needles, when the HIV gets in, it actually will not be able to enter their cells. So they won't ever develop an HIV infection inside of them, which is Crazy cool. This is one of the reasons why studying viral entry and viral replication is such a fascinating topic, because it allows us to fight these diseases that are so difficult to combat. HIV, of course, human immunodeficiency virus, targets your defenders, targets your white blood cells. And by targeting your defenders, it makes it very, very difficult to develop an active immune response to fight off HIV. So instead, HIV is oftentimes lethal in the fact that it does not kill the individual directly, but it brings your guard down, so that way there's no defense against other invaders. And so HIV weakens your immune system, so that way pathogens that historically wouldn't have been problematic now can be lethal. Kind of scary. But then, of course, just like with SARS-CoV-2, when the spike and the ACE protein bind, it gets in and replicates. Once HIV, its viral proteins bind to CD4 and CCR5 or CXCR4, the virus gets in and it begins to replicate. You also have the more typical bacteriophage. It's a T4 bacteriophage. So you may have recognized this drawing of a virus. Oops. Kind of a hexagon up top with spider legs down below. Those spider legs, or its viral tail, are the proteins that are the keys to entry into the bacteria itself. And so a T4 bacteriophage is a very specific type of virus that infects only bacteria. So not us, thank goodness. And then, of course, you have enveloped proteins like influenza that will get into the cell via membrane fusion. There is some components of still some membrane binding in terms of receptor-mediated membrane binding with membrane fusion, but ultimately it is slightly different in the fact that the membranes are fusing rather than endocytosis having to occur. I know it's a subtle difference, membrane fusion versus endocytosis, but it is very important in how we treat these different diseases. So those of you who are going to be pursuing a career in virology, keep that in mind. Know that there are differences in how we're going to be combating these different viruses based on the way that they enter. Now, the viral genome itself, as we talked about after entry, is going to need to be copied. And that differs quite drastically based on whether that viral genome is made up of DNA or whether it's made of RNA. Now, y'all have already had this section on DNA replication. The processes involved in copying DNA, including unzipping with helicase, stabilizing with single-stranded binding proteins, copying with polymerase 3 based on where primase put the RNA primers, and then connecting strands via DNA polymerase 1 and ligase. That process of DNA replication and DNA viruses, therefore, need to get into the nucleus to replicate. So DNA viruses are generally going to need to not only get through the plasma membrane to get to the cytoplasm, but then take an additional step to get into the nucleus, so that way their DNA can be replicated within the nucleus. Another benefit, quote unquote, to having a DNA nucleic acid core for a virus compared to an RNA nucleic acid core is that DNA nucleic acid cores generally have a lower mutation rate. So these DNA viruses are more stable over time. And whether that's a good thing or a bad thing depends quite highly. When it comes to our RNA viruses, we have RNA viruses that will replicate in the cytoplasm. And because they are replicating in the cytoplasm, they have, or because they are replicating as RNA in the cytoplasm, and RNA is inherently more unstable than DNA, these RNA-based viruses generally have a higher mutation rate. RNA viruses are also more common than DNA viruses. There's just more of them. And then we have a subset of RNA viruses that are what are termed retroviruses. Viruses that start with an RNA nucleic acid core, but then convert their RNA to DNA, and then from DNA, they can incorporate into your genome or make more copies of themselves within the cytoplasm. Lots of different angles on this, but ultimately, they do have ways to go from RNA to a DNA intermediate. Now, other trends to remind us of what are some of the differences between DNA and RNA viral genomes, and to break what we learned about DNA being mostly double-stranded and about DNA being double-stranded and RNA being single-stranded, is that viruses that have DNA or RNA can have either single-stranded DNA or double-stranded DNA, and single-stranded RNA or double-stranded RNA. So they have both. We also see viruses that have linear genomes, like adenoviruses, or circular genomes, like a lot of other viruses. And then sometimes they come in one fragment or many fragments. So you actually get this variation occurring both in DNA viruses and RNA viruses. And this table discusses some of the diversity of viruses that you might find of these different classes. For example, one of the most common dog viruses that we get our puppies vaccinated for is parvovirus. It is actually a single-stranded DNA virus. It converts to double-stranded DNA before the RNA is transcribed, and from the RNA, it can make the proteins that are its capsid and then continue to proliferate. You also have double-stranded RNA viruses, like the rotavirus, that transcribes mRNA from its just pure RNA genome. Again, with that RNA virus, RNA genome, you're going to get a higher rate of mutation, less stability. For other examples, we have what are called the positive and negative stranded viruses, RNA viruses. These are whether it is the actual protein coding region or whether it is the complement of the protein coding region, meaning that it is the other side. So which is which? Whether the plus side, which is the plus side, which is the minus side? Well, the plus side can act directly as mRNA. So this is your plus equals your protein coding. Your negative single-stranded RNA means that the mRNA is transcribed from the RNA genome. So you have to first go from your negative-stranded RNA to your mRNA, and then you can start copying everything. Hep C is actually one of these positive-stranded viruses that I think you're more likely to recognize than picornavirus, though it is one of the versions of common cold. The viruses that have reverse transcriptase, like HIV, copies itself into your genome, are incorporated into your DNA, making it incredibly difficult to get rid of because it then becomes part of your cells for perpetuity. That's pretty gnarly. And the mRNA that it makes is continually transcribed from your genome. So you become part virus? Well, good thing that your genome already had a whole bunch of virus-like sequence in those transposable elements, right? And we actually, there is a large train of thought that believes that the origin of all those transposable elements is actually from historical viral infections. So our genomes might be these large amalgamations of millions of years of viral infection happening. Kind of crazy to think about. And so just like you have RNA viruses with reverse transcriptase, you also have DNA viruses with reverse transcriptase, like hepatitis B. So let's go over a quick schematic of what the infection will look like all in all, so that way we understand from beginning to end what is happening with our virus. So, viral entry and replication looks something like this, with the steps being as follows. First and foremost, step 1 is attachment. The receptors or the plasma membrane on the surface of the cell and the surface of the virus will bind together. Step 2 is some sort of entry. Oftentimes, this is that endocytosis or fusion of the plasma membranes. Once the virus gets in, it then will release its genome, whether that be RNAs or DNAs, and ultimately get to this positive strand RNA to begin to make proteins. And this process of assembling new or making new features is called replication. And in replication, you are using the host machinery. Step 4 is after you've gone through making your machinery and you have synthesized a lot of proteins and synthesized more RNA, then you get to the assembly step, where new viral particles are being made. And then last but not least, you have the egress or exit of the virus. And the exit of the virus can be done in a couple different ways. We generally consider someone who has viral egression happening from their cells to be shedding the virus. And viral shedding is oftentimes that period of contagiousness. And viral shedding can happen in one of 3 ways. Either via budding. This is where the virus will actually just go against the plasma membrane on its own and bud out, taking some of the envelope with it. So this is fairly common for enveloped viruses. Second is exocytosis. This is where it actually goes into a vesicle, as pictured here, and that vesicle releases lots and lots and lots of viral particles. So in this way, the cell now becomes a repeating factory for viral replication. And then last but not least, apoptosis. This is where the virus just keeps copying itself within the cell up until the cell is completely depleted of resources and completely packed with virus to the point where it explodes. That sounds pretty violent, right? The cell exploding itself because it is so full. The apoptotic response is actually a little bit more complicated, and here's why I'm mentioning this fact. So viruses that replicate via apoptosis and exploding the cells of their host, well, they also cause the breakdown of the host genome and cause an apoptotic response. The apoptotic response recruits your macrophages, recruits the cells that eat your dead cells. Well, if apoptosis is recruiting the cells that eat your dead cells, the virus may be doing this process of apoptosis because the virus then wants to target the cells that eat your dead cells. This is actually the mode of action for HIV. It infects your macrophages by causing apoptosis to occur in your cells. Therefore, calling over the thing that they want to infect through apoptosis. If that's not clever, but at the same time cruel, I don't know what is. So let's get to some other vocabulary surrounding viruses now that we understand the process of their replication. Viruses are generally considered permissive because they need the host to replicate. They must be given permission in order to replicate. A productive infection is any infection that makes more viral particles. Meaning that it has to follow these steps: attachment, entry, replication, assembly, and aggression, or the shedding process, in order to make more of these. Whereas, as we mentioned earlier, you can have a viral infection that is not productive if you are lacking one of those points of entry, like in the case of HIV and that Ural Mountain population. An infection itself can lead to cell damage as your immune system is trying to identify cells that have been infected and destroy them. This destruction is referred to as a cytopathic effect. That destruction and that infection, as mentioned, can lead to either lysis, which is cell bursting, or budding, or exocytosis, as discussed above. Now, infections are viruses either follow a lytic life cycle, where the cell eventually will explode, or a lysogenic life cycle. And a lysogenic virus is one that copies itself into your genome and then will replicate later. This means that because it's not just always copying itself and is actually incorporating into your genome, that the virus and you have become one thing, to become one. But also that resting period introduces a time period of latency. Or wait period of the viral replication. Now, viral transmission can either be horizontal, meaning from person to person, or it can be vertical, from parents, like mother, to kids. Let's go ahead and, my handwriting is big, so let's go ahead and make this a little bit smaller here. Now, there are other types of infections or other consequences of infection, like hyperplasia, which increases cell mitosis. So cell division goes up. In plants, this will result in something called a gall, or a growth on a plant that is just a whole bunch of cells being copied over and over and over again, essentially a form of plant cancer. Just as there is a way to increase cell copying, there's a way to lower cell growth and division. We call that hypoplasia. Again, rather than a gall being symptomatic of hypoplasia in plants, abnormally low cell growth results in necrosis or dying tissues, dying spots on the plant, because the plant is not copying enough cells to keep that whole organ alive. Some animal-specific viral terms, or some terms that you generally find with viral animal infections, are an acute disease, is any disease that gets worse over short time periods. Oftentimes short time periods, but can also be quite long. A chronic disease is any disease that is long-lasting. An intermittent disease is any disease that has a latency period, meaning sometimes you have it, sometimes it seems not to be there. Herpes simplex virus is a good example of this. A disease in which you are asymptomatic is where you have it, you are shedding, but you have none of those cytopathic or cell-damaging effects, so you have no symptoms. Quite easily to spread when you are spreading it but don't even know it. And then the last is an oncogenic infection, a viral infection that carries a cancer risk. Obviously not so great. So the very last topic that we'll cover today in our discussion of viral diversity is actually not so related to viral diversity itself, but instead viral prevention. So what is the way that you can prevent from getting a viral infection? Well, the most common is a vaccine. A vaccine is an attenuated or weakened dead virus, or recently, thanks to developments with the COVID-19 vaccine, viral nucleic acids that expose the body, in particular the immune system, to the antigen or the identifying feature on the virus. In COVID-19's case, that's spike protein. So that future infections you can recognize that virus early and minimize the cytopathic effects. since this is so important, since vaccines are so important to modern human health, I'll go through that again. So a vaccine is an attenuated or dead virus or viral nucleic acids that are designed to expose your immune system to the antigen, which is whatever identifying feature on the virus that we need in order to recognize that it's in you, so that when you see it again and it's in its dangerous, not weakened, live form, you are able to recognize the viral infection early, plus have the antibodies at the ready to minimize the damage that the virus can cause inside of you. So the whole goal of this is to teach your immune system what the enemy looks like and what tool can be used to knock it out. Vaccines carry no risk of autism. I cannot believe that so many years after the retraction of the publication that provided a link between vaccines and autism, and the author of that study coming out and saying publicly many times that they fabricated the data out of whole cloth, completely made up, no connection, do we in society still have so many individuals who believe that vaccines cause autism? That is right. We still have people who believe that vaccines cause autism, despite the fact that the person who started this movement came out and said that they made up the data entirely. Yikes. Vaccines have side effects. But vaccines are designed so that way the side effects are actually less dangerous than the disease. You have side effects to your vaccine because you are putting in either weaker forms or dead forms or partially self-replicating, super dangerous parts of the virus so that your body mounts an immune response. Your body thinks you are sick with something dangerous, so it mounts a response as though you are, so you may feel sick, you might develop a fever, but those are all your body's responses to try to combat this foreign particle inside of you. That's normal, right? Like you're teaching your immune system is learning, and it can cause a fever or muscle aches or something of the sort. In the same way that when you train your lungs, you go for a run, and going for a run can cause the side effect of shortness of breath. It's not abnormal to get shortness of breath while running, because that's the point of running, to train your lungs. So that way later on you have less shortness of breath for similar activities. Just like a vaccine actually stimulates that immune response, so that way your immune system learns to fight in a safe space, and then when it gets exposed to the actual virus, it can destroy the virus before it can infect so many different tissues in your body. So vaccines do have side effects, but they are less dangerous than the actual disease. So not getting vaccinated does not make you immune from the side effects of or rather you do dodge the side effects of the vaccine, the normal immune response, but you are still exposed to the dangers of actually having the disease itself. And I can assure you that having a highly productive lytic disease that explodes many of your cells is much more dangerous to you long term than the vaccine. There is also no benefit to having an actual disease as compared to the vaccine. Most vaccines, and this is not true in every single case, but most vaccines are designed in such a way to try to replicate the life cycle of the virus so closely that it launches a similarly effective immune response. Again, this can be somewhat tricky, but for the most part, particularly with with the more recent COVID-19 vaccines, they do a much better job of approximating a COVID-19 immune response, so that way your body learns in the same way with the same efficacy as actually getting sick. And I included this statement "or better" because with vaccinations, like with the COVID-19 vaccine, uh, we were administering 2 doses. So that way your immune system learns the first time, does some antibody design to try to optimize the system the first time, and maybe if it's not perfect the first time, it then goes through that antibody design a second time with a second vaccine exposure, roughly 21 days after the first vaccine exposure, so that way you can really optimize your design. Your immune response would probably have a similar series of effects the first time around, but then you don't want to have to expose yourself a second time around just so that you can optimize your design. The vaccine gets rid of a lot of that risk. Now, there is some risk to vaccines. That risk that the vaccines carry is a risk of autoimmune disease. An autoimmune reaction is where your body designs antibodies for the virus, but in that antibody design, we think what's going on is that it also designs an antibody that happens to be similar enough to some of your own tissues that in fighting the virus, it also fights your own body. Now that's really bad to have an immune response to some of your own tissues. And autoimmune vaccines do carry a risk of autoimmune disorders. However, I do want to point out that not vaccinating and getting the disease still carries that risk of an autoimmune response. You can have an autoimmune response from a simple common flu. That type of autoimmune response is called Guillain-Barré. Ooh, let me make sure I'm spelling this right. Yeah, Guillain-Barré disease. And Guillain-Barré disease, or Guillain-Barré syndrome, can actually cause you to attack your nerve cells. So, in attacking a common cold infection, you can have temporary paralysis, and that's quite scary. This is why epidemiologists generally recommend that the portion of the population that does not carry a risk of autoimmune disease, which is a huge percentage depending on whether you're looking at, depending on the type of autoimmune disease you're looking at, but more than 75% of us don't carry this high risk of autoimmune disease, that if the vast majority of us get vaccinated, we can help not only protect ourselves, but also those folks who have some risk to the vaccination in the first place. It's not that the vaccine is dangerous inherently, it is that their immune systems are more likely to develop an autoimmune response with any infection. So those individuals who are at autoimmune risk are susceptible, more susceptible to autoimmune responses to any infection. So we want to just generally limit the amount of disease around them and around us, so that way they do not start attacking their nervous tissue when they meant to attack a cold. Much more work to be done on studying how autoimmune disorders arise, but know that that is where the concern around vaccines arises from, predominantly, but that the percentage of the population at autoimmune risk from vaccines is quite low. So please do discuss with your doctor or a rheumatologist, a autoimmune disorder specialist, if you have any questions on that particular front. But know that vaccines do not cause autism. So what are some, another potential concern is with vaccines that are live. These actually give living viral particles that have been weakened, but those viral particles can have back mutations. To be much more virulent. Mutations are random, it can occur, and in 2007, while we were administering the polio vaccine in Africa, there was a batch of it that did mutate back into the harmful form, and when we were vaccinating folks in Africa, we were actually infecting them with polio. That was a disaster. That was a horrifying human tragedy. Hopefully, we've learned from that and continue to monitor our live vaccines. But the good news is that most vaccines are not live, or a large portion of them are not live, and therefore do not carry this risk of back mutation. For example, the COVID-19 mRNA-based vaccines are not live vaccines, and therefore do not carry that risk. Outside of vaccines, what medications can we offer to help folks deal with viral infections? Well, some of those drugs are antiretrovirals that will actually stop the replication machinery for retroviruses. These drugs were first designed to combat HIV, which is a retrovirus, but have since been used in other type of viral infections that use similar machinery, which is kind of cool. And then the other one that I find really interesting are your nucleotide analogs. I've included an image of nucleotide analogs, like acyclovir, which is a herpes treatment, a herpes medicine, that has a similar structure in the nucleotide component, but has a different backbone. Rather than having a sugar backbone, it has a shorter backbone, but in doing so, the replication machinery that wants to copy the RNA viral genome will grab onto medicines like acyclovir and not be able to continue. So they gum up the works of viral assembly in the region. That's pretty crazy that we can trick these viral machines into grabbing onto something that they shouldn't, and then they won't really let go and slow down that spread and slow down the copying, so that way you get less cytopathic effect. That's super clever, at least I think so. The last two vocab terms are of two diseases that are not viral, but rather misfolded proteins, prions, and viroids, misfolded RNAs. Now these are particularly scary, particularly the prions, because they cause things like chronic wasting disease or mad cow disease, because they are a misfolded protein that when it bumps into one of your correctly folded proteins, it can self-propagate that misfolding and eventually cause neurodegeneration many years down the road. And there's really nothing that we can do about it other than make sure that the meat that we're eating does not contain these, and that we are not eating where it is more commonly concentrated in nervous tissue. So prions, if you needed evidence that these things are not living, that they're just trying to, they happen to be some motif that's self-replicating, look no further than prion diseases. They are just a misfolded protein that when they bump into another protein that's correctly folded, their chemistry causes that protein to misfold, and when that protein's misfolded, it'll bump into other proteins and continue to cause this misfolding. In the same way, or similarly, where viruses are just pieces of nucleic acid that try to copy themselves once they get into the right conditions, and they just keep copying themselves without caring what exactly they're causing to the host. They're just self-replicating, and that's all that they're doing. No purpose otherwise. Sometimes they don't cause much damage. In fact, you can have an asymptomatic infection. Sometimes you can be dead within 24 to 48 hours. Looking at you, Ebola. So we have, hopefully, a better understanding of viral diversity, where they land on the phylogenetic tree of life. They're their own tree. And from this, hopefully a better understanding of how to combat viral infections in your own life, and the importance of vaccines. So I'll see y'all next time when we actually start our exploration of all things living on our tree of life.